Okay, so get this. Today, we are diving deep into CRISPR.
CRISPR.
You know, that gene editing thing everyone's been talking about?
Right.
We've got a ton of your questions and articles and research on it, and frankly, I'm ready to get my hands dirty.
Yeah, it's exciting stuff. We're talking about tech that could rewrite the future of, like, everything. Medicine, agriculture.
Life itself, basically.
Exactly.
No pressure then. So where do we even begin with something this massive?
Well, I think we got to start with the basics.
Okay.
What CRISPR actually is.
Okay.
So imagine, like, a pair of super precise scissors
Okay.
that can cut and paste DNA that's CRISPR in a nutshell.
Okay. I'm picturing tiny scientists with even tinier scissors, but I'm guessing it's a bit more complicated than that.
Just a bit, yeah.
Yeah.
But the amazing thing is this whole groundbreaking tool was discovered almost by accident.
Really?
Yeah, Emmanuelle Charpentier, one of the scientists who made this breakthrough, she was actually studying bacteria.
Hold on. So the key to editing the code of life was hiding in, what, yogurt? Or one of those, like, glowing deep-sea creatures, or...
Even more intriguing, she was studying Streptococcus pyogenes, the bacteria that causes strep throat.
Get out. So the secret to gene editing was hiding in plain sight this whole time.
Kind of, yeah. It turns out bacteria have this incredible defense system called CRISPR,
Okay.
which stands for Get This Clustered, Regularly Interspaced, Short Palindromic Repeats.
You're telling me, but it sounds important. So what is this CRISPR system?
Basically, it's like an immune system, but for bacteria.
Okay.
So when a virus attacks, the bacteria can actually cut up the virus's DNA
Oh, wow.
and keep a little piece of it, like a mugshot,
Okay.
to remember it for next time.
So they're building like a DNA hit list of all the viruses they've encountered?
Right.
That's pretty smart for a single-celled organism.
Right. And that's actually where the Cas9 in CRISPR-Cas9 comes in.
Okay.
Cas9 is a protein that acts like the scissors, cutting up that invading viral DNA. And Charpentier's research, this is where she comes in, revealed this key molecule called tracRNA, a vital part of this whole system, guiding those Cas9 scissors.
Okay, so tracRNA is like the GPS, telling Cas9 exactly where to cut.
You got it.
Okay.
Now, this is where things get really exciting.
Okay.
Remember, at this point, they're just figuring out how bacteria do this.
Right?
But then comes Jennifer Doudna,
Okay.
a biochemist who teams up with Charpentier.
The classic scientific duo.
Right.
I love a good collaboration story.
Oh, yeah. And they had this aha moment in a cafe. Can you believe it?
No way.
Yeah. They realized they could recreate this bacterial system in a lab and get this actually reprogram it.
Wait, so instead of just cutting up virus DNA, they could make it cut any DNA?
Yes.
Anywhere they wanted?
Exactly.
Oh, wow.
They basically took a bacterial defense system and turned it into this programmable gene editing tool.
That is incredible.
It was, and still is, absolutely revolutionary.
No wonder they won a Nobel Prize for that.
Absolutely. 2012, that discovery was a seismic event in the world of science. And it all started with bacteria defending themselves from viruses.
Okay, so we've got bacteria, we've got viruses, we've got DNA scissors, we've got a Nobel Prize-winning breakthrough. I have to admit, I'm still a little fuzzy on how this Cas9 protein knows exactly where to cut the DNA.
Yeah.
It's like finding a single word in a giant library.
That's a great analogy, and it kind of highlights how crucial this next part is. Remember those mugshots we talked about, the bits of viral DNA the bacteria keep?
Right, right.
So imagine those mugshots are attached to these guide RNA molecules.
Okay.
The guide RNA with the mugshot then searches the entire DNA strand until it finds a perfect match.
Oh, wow.
And once it's found the matching sequence, the Cas9 protein gets to work, snipping the DNA right there.
So the guide RNA is like a detective carrying a wanted poster, searching for like a single bad guy in a huge city.
Exactly.
And once it finds the match, snip, the Cas9 swoops in for the arrest.
Precisely. And to make sure the Cas9 doesn't get carried away and start, cutting up all the DNA.
Right, right.
There's another layer of security here, the PAM sequence.
PAM sequence. That rings a bell.
Yeah, we touched on it briefly.
Yeah.
It stands for Protospacer Adjacent Motif, but we can just think of it as a short DNA sequence, specific code.
Okay, so what's so special about this PAM sequence?
Well, the Cas9 protein is actually quite picky. It can only cut the DNA if it finds this specific PAM sequence right next to the target sequence. No PAM, no cut.
So it's like a safety feature on those molecular scissors?
Yes.
Ensuring they only cut where they're supposed to?
Exactly.
But how on earth do scientists figure out the right PAM sequence for every single gene they want to edit?
Well, that's where things get really technical.
Okay.
For the CRISPR-Cas9 system that Charpentier and Doudna were studying, the PAM sequence is 5' NG3, where N can be any of the 4 DNA bases.
Right.
And you're right, identifying and understanding these PAM sequences is crucial for scientists because it basically dictates where and how CRISPR can be used.
So understanding this PAM code is like having the key to unlock CRISPR's full potential.
Yeah.
Like one of those combination locks, one wrong digit, and you're locked out.
Exactly. And just like there are different types of locks, there
Wow.
are different types of CRISPR systems out there, each with their own, unique properties and PAM sequences.
Okay, so maybe forget the tiny scientists. It's more like we're dealing with a whole team of microscopic locksmiths.
Yeah.
So we've got our microscopic locksmiths, our PAM codes, our guide RNA detectives. It's a whole world of molecular machinery in there.
It's pretty amazing stuff.
It really is. But let's talk about what all of this means for us, in the real world.
Okay.
Where are we actually seeing CRISPR make an impact?
Well, hold on to your hats because this is where things get really exciting.
Okay.
Let's start with medicine.
Yeah.
We're talking about the potential to cure inherited diseases that were once considered incurable.
Okay, now you're speaking my language. Give me the good news. What kind of diseases are we talking about?
Think sickle cell anemia, cystic fibrosis, Huntington's disease.
Right.
These are all caused by single gene mutations,
Yeah.
making them, prime targets for CRISPR therapy.
Okay.
Imagine correcting these errors directly at the source in the DNA itself.
Wow, it's like a genetic reset button.
Right.
No more passing these conditions down through generations. That's huge.
Yeah.
Are scientists actually doing this in humans yet?
It's still early days, but there are ongoing clinical trials with really promising results.
Okay.
For instance, there have been trials for sickle cell anemia, where they take patients' own blood cells, edit the faulty gene using CRISPR, and then transplant those edited cells back into the patient.
And it's actually working.
The results so far have been really encouraging.
Wow.
Some patients are producing healthy blood cells, essentially curing their disease.
Wow.
Now, it's still too early to call it a definitive cure,
Right.
but the potential is clearly there.
That's incredible. This could be life-changing for so many people.
Absolutely.
Are there any other areas where CRISPR is making waves?
Oh, absolutely. Let's move from the hospital to the farm.
Okay.
CRISPR is poised to revolutionize agriculture in ways we never even thought possible.
Okay. I'm intrigued because, genetically modified crops are already a thing.
Right.
So how is CRISPR different?
So you see, traditional GMOs often involve inserting genes from entirely different species like bacteria into plants.
Right.
With CRISPR, we can make very precise changes within a plant's own DNA,
Okay.
enhancing existing traits or even introducing entirely new ones.
So you're saying we can basically, enhance nature's blueprint,
Yeah.
making crops more resilient, more nutritious, without having to introduce foreign DNA.
You got it.
Okay.
Think of, crops that can withstand, say, drought conditions,
Okay.
resist pests and diseases, and even produce higher yields with less water and fertilizer.
Wow. So CRISPR could be a major player in tackling issues like climate change and global food security.
Absolutely, yeah.
That's a game changer.
It is.
We're talking about feeding the planet.
Exactly. And we're already seeing some amazing examples of CRISPR-edited crops. There's rice that's engineered to absorb less heavy metals from the soil,
Okay.
making it safer for consumption in areas, with heavy metal contamination.
That's brilliant. Safer food, more resilient crops. It's like CRISPR is a superhero for our planet.
It has that potential.
It really does.
And these are just a couple examples. We're only just beginning to scratch the surface of what CRISPR can do.
Right.
There's also groundbreaking research happening in using CRISPR for, say, diagnostics, gene therapy for cancer, and even, get this, engineering mosquitoes
Yeah.
to combat diseases like malaria.
It's almost hard to wrap your head around all the possibilities.
I know, it's a lot.
But with a technology this powerful, I have to imagine there are some pretty big ethical questions that come with it.
Oh, you're absolutely right. While the potential benefits of CRISPR are immense, we need to be mindful of the potential risks and ethical implications.
Okay, so let's talk about the elephant in the room we're talking about, literally editing the code of life. Where do we draw the line?
That's the million-dollar question, isn't it?
Yeah.
One of the most hotly debated issues is the idea of germline editing.
Germline editing, so that's editing embryos, right?
Right.
Making changes that would be passed down to future generations.
Exactly. And that's where things get ethically murky because on the one hand, you have the potential to eliminate inherited diseases from entire family trees.
Right.
But on the other hand, you're making changes to the human gene pool that are irreversible
Right.
with, potentially unknown long-term consequences. It's a heavy responsibility to bear.
It's like we're holding the future of humanity in our hands. How do we even begin to navigate these ethical dilemmas?
It's a complex issue with no easy answers.
Yeah.
There are scientists, ethicists, policymakers, the public, all grappling with these questions right now. Open dialogue, transparency, and, carefully crafted
Right.
regulations
Yeah.
will be crucial in ensuring CRISPR is used responsibly.
Right. For the benefit of all humankind.
Exactly.
Okay, so we've talked about the amazing potential of CRISPR in medicine and agriculture, but also the ethical tightrope we need to walk.
Right.
Is there anything else about CRISPR that we should be aware of? Anything we haven't covered yet?
Yes, actually, it's important to remember that CRISPR-Cas9, while groundbreaking, is not the be-all and end-all of gene editing.
Wait, there's more. I thought CRISPR-Cas9 was the cutting edge.
It certainly was a major leap forward, but the field of gene editing is constantly evolving.
Oh?
Remember all those different types of bacterial defense systems we talked about earlier?
Right, yeah.
Scientists are now discovering and engineering new CRISPR systems with, even more intriguing properties.
Okay, now you're just teasing me. Give me the scoop. What else can these new CRISPR systems do?
Well, some of these systems have different PAM sequence requirements, which means scientists can target a wider range of genes with even greater precision.
Okay.
But here's where it gets really interesting. There are CRISPR systems that don't even target DNA at all.
Really?
They target RNA.
Hold on, RNA. As in, like, the trusty sidekick to DNA.
Right.
What's so special about targeting RNA instead of DNA?
That's a great question. So remember, DNA is like the master blueprint, right?
Right.
And RNA is like the messenger carrying out those instructions.
Right, like the mail carrier delivering genetic instructions throughout the cell.
Exactly.
Right.
And so by targeting RNA instead of DNA, scientists can temporarily tweak gene expression without making permanent changes to that underlying blueprint.
Okay, so instead of editing the original blueprint, we're intercepting the messenger and, like, changing the instructions along the way. That's incredibly clever.
Nature is full of these elegant solutions, isn't it?
It really is.
And it opens up this whole new realm of possibilities for CRISPR technology, right?
Yeah.
Imagine being able to fine-tune gene activity, almost like a dimmer switch, turning it up or down as needed without permanently altering the DNA itself.
So we could potentially treat diseases that are caused by overactive genes.
Right.
Or boost the expression of genes that are, underperforming.
Yeah.
All without making permanent edits to the DNA.
Exactly.
That's incredible.
Yeah.
It seems much safer than messing with the original blueprint.
That's the idea. It's like a temporary intervention
Right.
rather than a permanent rewrite. And it allows for more control and potentially reduces the risk, of unintended consequences.
This is mind-blowing. It feels like every time we uncover one layer of CRISPR, there's like another layer of complexity and potential underneath.
It's true. CRISPR isn't just, one technology.
Okay.
It's a rapidly evolving field, and there are new discoveries happening all the time. Who knows what incredible tools scientists will uncover next,
It's both exhilarating and a little bit daunting to think about, isn't it?
It is.
The future of gene editing is brimming with possibilities.
It certainly is. And as we continue to explore this new frontier, I think it's more important than ever to engage in these, thoughtful discussions about the ethical implications of this powerful technology.
Absolutely. We can't just focus on the can we without grappling with the should we.
Yeah.
It's a conversation that needs to involve everyone, not just scientists.
I agree. The choices we make today about how we develop and utilize CRISPR technology will have a profound impact on generations to come.
We're talking about shaping the future of life itself.
Exactly.
No pressure, right.
Right.
But on that note, I think it's time to start wrapping up this deep dive.
Okay. Sounds good.
We've covered a lot of ground, from the intricate workings of CRISPR-Cas9 to the vast potential of this technology in medicine, agriculture, and beyond.
Yeah, it's been a really fascinating conversation.
It really has. You came armed with some incredibly thought-provoking questions and research.
Well, thank you.
And I really hope this deep dive has given you even more to ponder.
Me too.
And for anyone listening, if this conversation has sparked your curiosity, don't let it end here. Dive into those articles you've been saving, do some more research, and keep asking those tough questions.
Yes, because the future of gene editing, and perhaps even humanity itself, depends on informed and engaged citizens like you.
Beautifully said. So until next time, keep exploring, keep questioning, and keep those brains buzzing.